FSU Inventors and some MEDC grant recipients. And that will be Eric Nybo, Sonali Kurup, and Danielle Taylor, I think is going to kick us off.
Great. Everybody see my screen okay? Super. So thank you for having me here. My invention is Protoconch. That's our prototype name. It's a combination of prototype and a conch, which of course means seashell, because we're speaking here about the inner ear, the workings of the inner ear. It is a cochlear implant spectacle solution, and we'll get more into the details in just a moment. I do want to say a quick word about some of the people who were involved in the early part of this, because I didn't have put those in the PowerPoint. But when this idea first came to mind, I reached out to engineering technology, specifically Luke Hedman, who I believe was one of the people who actually spoke even before our introduction today at 8 o'clock. He is a professor over at engineering technology and runs one of the capstone courses, I believe the capstone course for one of the programs over there, where their very promising students are given innovative ideas and asked, make it a reality. He hooked me up with one of the students there, Jacqueline Vanderplug, who is now a alum of the program and working in private company. But she was able to work with me and come up with the germ of the idea, come up with a few very basic prototypes that we have taken from there. So great thanks to Professor Hedman, to Jacqueline, who I don't believe it is on this call, but it wouldn't have happened without them and without the collaboration between our college at MCO and engineering technology as well. So let's get started. The problem that we were trying to address is that some people have behind the ear cochlear implants. Those are cochlear implants that, as you can see, sit behind and on top of the auricle of the ear. These people can have trouble wearing spectacle lenses at the same time as they wear the devices that allow them to hear anything. Because spectacles, as you can see, compete over the same space. They physically interfere with CIs. So what problems are we talking about here? Well, security problems. People about my age who might need to wear reading glasses. Of course, anybody here who wears reading glasses knows you need to take them off and put them back on again throughout the day. And if you're doing that often, as you're sliding them past this very important piece of equipment that has a microphone in it and processes sound and then sends it through the wire to the coil, where it transmits across the skin into a receptor that's been surgically implanted that then connects to the inner ear so that you can hear. That's the way a cochlear implant works. Very basic. I'm sure it's much more complex than that. But you can understand how that would be a problem throughout the day. And so there are a lot of solutions people have had to try to address this problem, but very few of them work well. So security is one issue, but also for kids. A lot of children who are born deaf, like my 3-year-old son, who have bilateral cochlear implants, if they want to wear sunglasses because it's bright outside, there are certain conditions, like my son has Waardenburg syndrome. Fortunately, the only part of that that's out there is hearing loss and light sensitivity, because his eyes are very depigmented. And so as a result, when he goes outside, sunlight that doesn't bother me bothers him. Well, he'll want to wear sunglasses later in life, but it's difficult, particularly for a child who has limited dexterity, to be able to do that effectively. And then too, there's stories about people who have gone to all sorts of different extents to try to mitigate this, and they find a way to improve the security, but that decreases the comfort. And you have situations where kids are coming home from school, for example, who have had some sort of a homebrew connection with tape or with rubber bands, say, and then they've got a sore on the side of their scalp, right there above the ear. We want to see if we can find a way to address this. So that's the problem we're trying to address. And here's what we came up with. Protoconch. It is currently patent pending. As you can see, it is a connection that attaches to the end of the behind the ear cochlear implant processor and then attaches to the temple of the glasses. So this allows for them to essentially become one unit, where the behind the ear part of the cochlear implant acts as the securing back mechanism of the temple. So that one, we have something that is secure. 2, we have something that is comfortable. And 3, you can't really see it in this drawing because this is an early prototype, but 3, it also allows the glasses to be removed and put back on easily throughout the day without disturbing the cochlear implant. So now the two elements are no longer competing for the space over the ear. So, of course, the value helps the CI user who either must wear or wishes to wear. Like, for example, in the case of fashion, I mean, there's no reason why someone who has CIs should not be able to wear glasses for fashion. Some people do that. And also sunglasses, like we talked about before. So the person who must or wishes to wear spectacles by providing that secure, comfortable connection between the two elements, as well as being more facile, being able to remove and put the glasses back on as needed throughout the day. As we talked about, existing alternatives are bulky, unstable, and difficult to adjust. This helps a lot of people, not just the patient. When we're talking about family members, caretakers, particularly with young children, it helps them with their peace of mind. Teachers can be involved because a lot of the teachers who are involved in oral deaf schools, this is a concern. How do we keep things stable so when the children are playing, their CIs aren't falling off? Therapists, speech-language pathologists will be very interested in this. I've spoken to some of them. They think this is exactly the kind of option we want to be able to provide for our patients to improve their quality of life. Audiologists as well. And then the hope is that we can actually talk to one of the CI company reps and perhaps even get them on board for a licensing sort of a decision or something like that. That's our hope. We really feel that that's the best way to get this to people, is by getting one of those big companies involved and adding it to their armamentarium of different accessories that they use to improve the quality of life of their patients. The market opportunity is small. I won't go into all the details here, but when you look at all the people who have behind the ear CIs worldwide, you couple that with the fact that we're probably looking at the English-speaking world, at least at first, and then we're really focusing in a target market on those people who must also wear glasses because they have reduced vision without them. We're probably looking at a very small number at first, 10 to 30,000. And if you're looking at about $30 per unit, though our market research indicates we could go as high as 75, but we're not trying to get rich on this thing, we're trying to help people. So if we're looking at $30 per unit or so, you're probably looking at yearly revenues and sales of $15,000 to $60,000. Again, nobody's getting rich off this, but that's one of the things that's so cool about working at Ferris, right? I don't have to make a big monetary impact in order to make an impact, because every individual person that benefits from something like this, it's a huge impact to that person and to his friends, his family, his loved ones, his doctors. So if we make some money, great, but ultimately it's about getting that out there so people can have an improved quality of life. Competitive analysis, there are a few different options. Kanso and Neptune are CIs where the processor sits back here as opposed to on top of the ear. I'm sorry, I'm pointing to myself and I'm just a little box on your screen. But basically the processor connects to the skull, the scalp, back behind the ear. And those are options more people are using. Not everybody uses them though. There's still a substantial number of people who use the cochlear implants that sit above the ear. The home fix ideas like tape and rubber bands, again, they kind of work, but there's a lot of problems with security, with comfort, things of that nature. So Protoconch addresses all of these potential concerns on the competitive analysis. I won't get into regulation very much, except to say that this will probably be classified as a minimally invasive or minimal risk device for FDA. We will need to go through an FDA approval process of some kind, almost certainly. And so we're still going to have to consider funding for that once we get through our beta testing protocol. But for now, we're sure that this can be classified as an accessory of a more advanced technology like the cochlear implant. Fleshing out the idea. Well, we had the idea. We worked up the prototype with engineering technology. Since it was invented at Ferris, of course, we submitted to the Office of Research and Sponsored Programs and IP and Tech Transfer. They walked us through the process. They said, hey, we've got this connection with the innovation network statewide. Let's submit to Michigan State University and have them do an analysis. And that's where I came in contact with Denise and other people in the EDC. They gave us an opinion. They said, basically, yeah, you're not going to make a lot of money on this, but it's probably useful. And then they put us in contact with people at University of Michigan who enrolled me and our program in the fast-paced program. And the idea with fast-paced is that it took this idea, one, asked the question, Does it have any commercializable potential? Yeah, it's a cool idea, but is anyone actually going to buy it? There's thousands of cool ideas. What actually is going to be useful? So we were able to do an analysis and find out, yeah, in fact, with our customer discovery, this seems to be very the kind of thing that people who are involved by this are very interested in. We spoke to over 30 people. We're going for 50, but COVID happened literally in the middle of our customer discovery, which threw a bit of a wrench in the works. But what we find is that the need for this kind of connection is well perceived. Patients and families are willing to pay up to $75 if the device works well. There's some interest from the spectacle or cochlear implant industry, but it's difficult getting in touch with representatives once we have a product that hopefully will be easier. And even that this could be the kind of thing, if we classify as a medical device, that could be reimbursable by insurance, because we can make the argument, particularly for children who must wear glasses, that this has a developmental benefit. It helps their vision and their hearing develop by preventing them from losing their ears, essentially, their cochlear implants, and their glasses throughout the day. From there, we received a grant, a generous grant from Michigan College of Optometry's Vision Research Institute so that we could apply for patent protection. And then we also received a very generous grant from MEDC's Advanced Proof of Concept Grant Fund to fund our prototyping, which we're currently in. You can see there's our first prototype. The model, it's who we had, I'm sorry. But you can see the connection early on. There's still some things to work on. The connection between the cochlear implant and the glasses needs to be secured a little bit. We've made a few additional adjustments. We're also working on a reconnector that would allow you to reconnect the cut end of the glasses, because it is necessary to snip the temple in half to use this. It would allow somebody, if they're not wearing their cochlear implants, but at the end of the day, say, but want to wear their glasses, to reconnect so they can wear their glasses securely, even in the absence of their cochlear implants. So we've got a really exciting little piece that'll allow us to do that. So right now we're finishing up Alpha 1 prototyping. The hope is we can develop our Beta 1 prototype by April 1st. That's a little ambitious. It may be more like May 1st. But once the beta prototype is there, then we can start a commercializable version or develop a commercializable version through beta testing. So that's the general idea. Protoconch addresses a specific felt need among CI patients and parents who are generally enthusiastic about it. Commercial form is currently in alpha prototyping, showing early promise. We want to begin and complete beta testing as soon as possible. And of course, we still need to secure funding for de novo accessory regulation from FDA, but that's another step after we're done with this one. And the ultimate goal is licensing to a medical device or cochlear implant company. That's our preference. We can make this ourselves, but of course, we're going to be able to have more of an impact if we license. So thank you for your time, and I believe we'll take questions at the end once everyone's had a chance to present. So thanks again.
Yeah, thank you, Daniel. We'll do questions at the end. Eric, would you like to go next?
Oh, sure. Sonali, is that okay with you?
Yes, that's fine.
Okay.
Well, you need screen share capabilities. I think Jonathan may be able to give you that.
I do have screen share capabilities. I'm just going to pull up my slide deck here. I pasted my patent information for the invention I'll be talking about today in the chat. Not really talking so much about the inventor track for this, but I am talking about work that has spun off since we were awarded our MEDC Advanced Proof of Concept Fund a couple years ago. So I pasted in the chat. Our patent was actually just allowed. It will be issued in March. So we just actually paid the issuance fee yesterday. So that's very exciting. So kind of showing what happens at the end of sort of the patent process, and our partners at MEDC were critical for that. So it's kind of a nice comparison, you know, have all these inventors at different stages of the invention process. So my invention has taken on an acronym. Acronyms are accessible to people. So I call my invention BIOPOLYMER, which is short for BioBricks Polyketide Metabolic Engineering Platform. You can see the acronym is capitalized throughout there. And when I tell that to people, they say, well, just what in the heck is biopolymer? What is the problem you're trying to solve? So we work with drugs. I'm a professor here in the College of Pharmacy, and so my focus is on medicinal chemistry of anthracyclines. Anthracyclines are among the broadest spectrum chemotherapy agents. They're used to treat a variety of cancers, breast cancer, ovarian cancer, all the way to liquid tumors such as leukemias, non-Hodgkin's lymphoma, et cetera. So they've been widely respected in the literature. They've been used clinically for over 50 years, and they are topoisomerase 2 inhibitors. And so what they do is they inhibit the DNA replication of quickly growing cancer cells. But they've been used clinically for 50, 60 years, so they're well appreciated. They have exciting anti-cancer and antibacterial activities. So these drugs are actually produced by soil-dwelling microorganisms called streptomyces, which I'll be talking about during today's talk. Bacteria produce these as antibiotics to kill off other bacteria to compete for resources. But since the drugs are so toxic in people, we have to use them as anti-cancer drugs. We have to use them to treat probably one of the most dire indications that we have, which is cancer. So we use these in people as anti-cancer agents. The problem with these agents, though, is that they're very, very cardiotoxic. So patients that are on anthracyclines can only stay on them for so long. So they have a lifetime maximum tolerated dose of like 500 milligrams per kilogram squared. So what we want to do is we want to develop better analogs that are better tolerated in terms of the cardiotoxicity. And with some of my collaborators, we believe that there's actually structural changes you can make to the drug that can have the beneficial aspects, which are the cancer killing aspects, but can actually dissect the cardiotoxicity from them. We actually think a lot of the cardiotoxicity is due to the fact that these do bind to topoisomerase 2. What we want to find are analogs that actually are involved in histone eviction. And we think it's the histone eviction activity that is more beneficial to the cancer patient. So if we can create analogs that have histone eviction capabilities, but reduce the cardiotoxicity, we think that we might be able to usher in a new generation of analogs. And so my project really uses the bacteria that produce these molecules in nature to produce the new analogs. So we reprogram the bacteria to make new anthracycline compounds. And the goal of my NSF-funded project is to use something called combinatorial biosynthesis, where we mix and match enzymes from different pathways. And these enzymes are called polyketide synthases. And so the goal of the project is to mix and match selected polyketide synthase enzymes with post-polyketide synthase enzymes to produce productive biosynthetic pathways that will rationally produce the designed anthracyclinones. So we have a bunch of enzymes that we're going to recombine in interesting ways to hopefully make new analogs. The overall objective of the project is to characterize different PKS genetic parts that will form these new in vivo biosynthetic pathways to produce known and new to nature anthracyclinones. And so really the goal of biopolymer is to enhance access to natural products. A lot of the analogs that we're working on would be very difficult to synthesize chemically, aren't available commercially. And so we believe that we're creating a product that would help drug companies or customers get access to these natural products much more quickly. And the project has two specific aims. The first specific aim, and I'm not going to get too deep in the weeds on the chemistry here, too deep in the weeds on the biosynthesis. We don't have time. But really specific aim in one and two are about characterizing genetic parts. So we're going to characterize genetic parts to make our pathways work. And I guess the best image I can give you of biopolymer is of a craftsman toolbox. What we're working on is a toolbox, and biopolymer is composed of genes. Genes encode gene products that are enzymes that will compose these pathways. So the first thing we work with are these biosynthetic genes, building blocks. Then we have promoters, and promoters are like genetic on switches. So by using promoters, we can induce transcription of our genes that will then go on to form enzymes that build out the pathway. So promoters are on switches. These are very important building blocks that we work with. The third thing that we work with is streptomyces. Streptomyces is the organism that natively produces these. It's a very competent cell factory that if we put the genetic blueprints into streptomyces, we can kind of build out our pathways. And over here, we have a picture of streptomyces growing on a plate. These blue droplets are actually antibiotics. You can see them in the plate. It's really fascinating. It's producing so much of this blue antibiotic called actinirodin that it's actually blebbing up from the plate in these little droplets. So Streptomyces is the chassis or the host. We have terminators. Terminators are like stop signs that stop transcription. And so our pathways, our genetics reads a lot like a sentence. So we have a promoter, which starts the transcription. We have genes that are then transcribed, and then we have off switches that shut off the transcription to make this very efficient. And the last thing that we have are vectors, and vectors are the means of introducing the DNA into Streptomyces. So that's kind of like the parchment paper that you put the blueprint on. So these are all the components of biopolymer. And again, I only have 20 minutes today, but I want to give you an update of what we have done in terms of what is the scope of our biopolymer toolbox. To date, we have synthesized many new BioBricks parts to engineer production of anthracyclinones. To date, we have over 46 unique polyketide synthase genes that can be recombined in interesting fashions. We have 13 post-polyketide synthase tailoring genes. We have 8 unique promoters that occur at a variety of promotion strengths that can be used to drive expression. We have 3 unique terminators, and we have 5 unique vectors that can be used for the metabolic engineering of Streptomyces. And this toolbox is probably one of the largest of its kind for type 2 polyketide biosynthesis. So I'm excited to tell you about a recent engineering stress test we did with our collaborators in Finland. And so I'm happy to show you the results of that stress test. But what this shows is that we're collaborating with labs across the world, and they're able to use our toolbox to do biosynthesis, which is fascinating. So the star of the show is Streptomyces silicolor, which was producing that blue-colored antibiotic that you saw before. Streptomyces silicolor, again, grows in the soil. It's a gram-positive microorganism, and it forms these very interesting spore chains. And so it's a lot like fungus in a way, because it makes these spore chains, and it kind of grows out from there, and it forms these spores that you can see on plates. And we call Streptomyces nature's antibiotic factories because a lot of the antibiotics and anti-cancer drugs we use come from this genus of bacteria. So erythromycin A, tetracyclines, doxorubicin that I already talked about, rifampicin, tacrolimus, all of those are well-appreciated drugs that are used as immunosuppressants, anti-cancer drugs, antibiotics that are used in the clinic. And they come from this genus of bacteria. The bacteria that we use, though, are not this one. Sorry, my slides are going crazy. We use superhosts, and superhosts are actually not the blue antibiotic producing bug, but they are bacteria that have been genetically engineered to lack the production of biosynthetic gene clusters. And so by using these superhosts, we can actually take out the blueprints for the antibiotics they were making, and we can transplant new instructions into them to reprogram them. And that is the process that we use to kind of engineer these pathways. And the most important aspect of what I can convey to you, aside from what biopolymer is, the toolbox concept, is the design, build, test, learn cycle. This is a key concept in engineering where you design an experiment, you design a pathway, you design a host with the right modifications. You build the pathway through DNA assembly and gene synthesis. Then you do testing via growing up the microorganisms, extracting out the compounds, and doing chemical profiling. And from that, you learn how successful your plan was. And then from that, you go through another iteration of the design, build, test, learn cycle. And so today, I'm going to take you through a stress test of this entire process. So this work was actually done in collaboration with my good colleague, Professor Mikko Metsäkettälä from the University of Turku in Finland. And it was his graduate student that actually put these genes together and did the work that I'm talking to you about, but it's an illustration of how biopolymer works. So our target was this molecule down here. We had a tetracyclic molecule called naglomycinone that we wanted to synthesize at the end of a long 10-step process. So this is a 10-step biosynthetic pathway that we said, okay, we're going to use our best guess on how this pathway should be put together. We're going to put together 10 genes and we're going to go. We're just going to see if we can make naglomycinone. And I'm going to show you the results of how that stress test worked out. As it turns out, we didn't end up with naglomycinone. We ended up somewhere around here. So we're 7 steps in, which actually isn't terrible. But the way I try to tell people about metabolic engineering, it's like a pipeline. And so what we found out is that our pipeline is very strong here. We get a lot of these early intermediates, but we only get a smattering of the smaller ones. And so what we found out was we have a very large pipeline at the start of the pathway, but very quickly that pipeline becomes very small. And I know you're looking at me through a camera right now, like Dan was saying. So imagine your early pipeline is 10 feet wide, and then it suddenly narrows to about 2 inches in diameter. And that's kind of what we're seeing. We don't actually see naglomycinone, but through metabolomics, we were able to detect several intermediates in the pathway that tell us how well the pathway is working. So through metabolomics, we were able to identify several shunt products that the pathway is producing. Decarboxy anhydronagalonic acid, S25O2, SEC43 and SEC15, all of these are published metabolites that are known to be produced by polyketides. And using mass spectrometry, we're able to identify them. And so by profiling the metabolites in the pathway, we can see how well the pathway is working. And that allows us to learn from the process to start the next phase of the design, build, test, learn cycle. So we detected 5 metabolites that were produced in reasonable quantity and a couple others that we could just sort of barely detect. So the largest metabolite was this one, SEC15, but we had a smattering of others that were tricyclic, like decarboxy anhydronagalonic acid and S25O2. And then down here towards the end of the pathway, we had barely detectable levels of what could be our target molecule here, naglomycinone, and the penultimate metabolite, nagalavacetone. And so this is looking at the UV-Vis spectra of the different metabolites. The ones that we're interested in have strong absorbance at 430 and 440, which we can only detect a couple of these. The earlier metabolites that are monocyclic and dicyclic have absorbances more at the 290 level. So again, this is just kind of showing all of the metabolites overlaid with each other, showing that we had a couple that are the intended, but a very low level of them. And so from this analysis, we were able to tease out 5 unique metabolites that were produced in this initial stress test: decarboxy anhydronagalonic acid over here, SEC15, which this is the major metabolite of 80% of the metabolites are this one, SEC43, which is also pretty major, and then finally nagalonic acid methylester, and S25O2 was another minor metabolite. So again, we've gone through 1 step of the design, build, test, learn cycle. So where do we go next? Well, the first thing we have to do is we have to balance the pathway. The later steps are not as well transcribed. There's not as much of the enzymes to push the carbon flux down the pathway. Secondly, by changing the host strain, we can change how well the pathway is utilized. So by escalating to a better host strain, we might get better pathway balancing. We might have more higher enzyme levels that would allow us to move this forward. Another way to kind of look at this would be quantitative real-time PCR. So the first question you might ask is, are our genes being transcribed? This is kind of a secondary thing we can do in addition to the metabolomics. And the most important thing we can always do is design, build, test, and learn. So my highest acknowledgements are for my students. This work is done all by undergraduates here at Ferris. The work I just showed, this was done by Rongbin Wang from the Miko Metzaketala lab, but on my side, I work completely with undergraduate students: Caitlin Brown, Jennifer Wen, Nina Gorjajewska, Mitch Lukin, Shannon Hunt, and Kennedy Rebschlager. I acknowledge the National Cancer Institute for an R15 grant. I acknowledge the National Science Foundation, the CBET division for funding. And I also would like to acknowledge the MEDC. If the MEDC hadn't funded our project approximately 3 years ago, none of these other follow-on projects would have been possible. I also want to thank Dr. Felix Amisa, Dr. Tom Dowling, Denise Graves from MEDC, my external collaborators, Maden, Miko, Hongbing, and certainly I want to thank the Ferris State University College of Pharmacy Alumni Advisory Board for their support as well in the form of an instrument for this project. We have some of the most engaged alumni here at Ferris State University. It's truly awesome to work here, and I'm excited to hear from Sonali and to get your questions later. So thank you for the time.
Thank you, Eric. Sonali?
Yes, I'm going to share screen now. All right. Thank you, everyone, for joining us today. It was a wonderful talk from Dr. Nybo as well as Dr. Taylor. I'm going last. I hope I continue in the same trend as the two of them before me. I'm Sonali Kurup. I joined Ferris State in 2018. I started my academic career at Roosevelt University before that. And some of the data that I'm presenting here was generated partly when I was at Roosevelt, but most of it was generated at Ferris. I've also limited my talk to only the data that served as prelim data to my advanced grant, and then what I'm doing on the advanced grant. I do have additional funding, and I guess I'll say that at the end, but none of that data is included in here. I've just limited it to the advanced grant because I think that was the biggest stepping stone for me that took me into a bigger league. So I'm going to start out. I work in the cancer research area. I am a medicinal chemist, similar to Dr. Nybo. My research focuses on the design, synthesis, and evaluation of small molecule EGFR AURK inhibitors for multi-resistant lung cancer. And they're basically dual targeted molecules that inhibit EGFR, epidermal growth factor receptor kinase, and aurora kinase. With respect to, before I get into the project, I just wanted to say a little bit about what my research lab does. So I guess it should be titled Kurup Lab more appropriately, but I just thought research focus would apply just as well. So I am a medicinal chemist, like I said, and I am a small molecule synthetic chemist. And so most of my work involves structure-based drug design. So I look at enzyme active sites and enzyme pockets and design molecules for those pockets, optimizing binding interactions. I then go on to synthesize those molecules in lab and then evaluate them. And so the evaluation at this point has been mostly either collaborative or through contract research organizations. But my strengths and skills are in structure-based drug design and synthetic chemistry. I do have some experience with enzymatic assays as part of my postdoctoral training at U of M, but I haven't had an opportunity to set that up in lab yet. So with that, I'm going to go a little bit more into the project. I work on multi-targeted kinase inhibitors. That has been my focus since graduate school. So originally, I've always worked on epidermal growth factor receptor kinase. It was one of the first few drugs after imatinib that got approved amongst the targeted agents for cancer. And so the first drug in there was gefitinib, and then the second one immediately after was erlotinib. But then resistance, those were the first generation drugs, but resistance developed rapidly. And so it's always been an area of intense research interest. A target that I am looking at to overcome resistance to EGFR is aurora kinase. And so I'll go a little bit more into what these two targets do in subsequent slides. I just wanted to introduce it here just so that you all know that this is my main focus. Again, like I said in the beginning, these molecules that I make in lab are designed as anti-cancer agents. And so they will help overcome cancer and inhibit cancer cells and inhibit tumor growth. And they are primarily focused, instead of just general lung cancer or general gastric cancer where my compounds have applications, they are more, I think the niche area would be multi-resistant lung cancer because there are really no drugs that treat or have been approved thus far for mutant EGFR positive cancers as well as mutant KRAS positive cancers. So there's a lot of research in these two areas, but there are no drugs that have been approved. And so I don't have a nice market need slide like Dr. Taylor, but that's my niche. So I'm just saying that out here. So a little bit about EGFR. So EGFR is epidermal growth factor receptor kinase. So it is a kinase. And so if you all think back to when you all had biochemistry, if you had biochemistry, a kinase transfers a phosphate group onto residues, amino acid residues in substrate proteins. And so EGFR is a receptor tyrosine kinase. So it transfers a phosphate from ATP, which is your phosphate source, to a tyrosine residue in your substrate protein, forming a phosphorylated tyrosine and ADP at the end as the end product. So most of the efforts when they've designed EGFR inhibitors have focused on so most of the drugs that were initially approved focused on looking at the kinase enzyme itself. And so they focused either on the substrate protein or the ATP pocket. Now, if you look at ATP, it is a small molecule itself, so it's already drug-like. It was much easier to design a molecule that would mimic ATP, and so that's where most of the drugs came from. So most of the drugs that are approved are ATP competitive, small molecule EGFR inhibitors. A little bit more about my background. I did work on targeting the substrate protein site as well, but those are very difficult to make, and they're also incorporating drug-like properties has been hard, but that still remains an area of interest, even though that's not the focus of this talk. A little more background, there are erlotinib, gefitinib were the first generation drugs that were approved, and then afatinib, osimertinib are second and third generation drugs that came out later to help overcome some of the resistance. Now, when I spoke about EGFR, again, this is just a cartoon just to make it a little simpler to understand the background before I get into my main project. EGFR has an extracellular domain, a transmembrane domain, and an intracellular domain. Most drugs, so ATP is found, or the kinase portion is found on the intracellular domain, and that's where the small molecule inhibitors that have been approved target. That's what they target. There are also monoclonal antibodies for EGFR that target the extracellular domain. I am not looking into that, and those molecules are not effective in lung cancer, which is the area that I'm focusing on. We are looking mostly at the ATP pocket, which is on this intracellular domain. Now, the reason I put this slide here is because there are problems. So why am I working in this area if there are already drugs that are approved for it? And so the main reason I'm working in this area is because there is a problem of resistance, like I said, and one of the main mechanisms for resistance is a mutation in the ATP pocket. And so you can have a double mutant, which is L858R T790M. That was the original mutant that came out, and it is a gatekeeper mutation, which prevents the first generation drugs, erlotinib and gefitinib, from binding. This then resulted in novel generations of EGFR inhibitors. However, that has now resulted in developing a third mutation. And so now you have a cysteine 797S mutation. And so right now, there are no drugs that are available that can overcome resistance or in patients who express this form of EGFR, which is the triple mutant. And so there is an unmet need, and there's a lot of research that is still occurring, and I'll come into what is happening and how my idea competes with that. Another mechanism of resistance involves downstream signaling. So I didn't really go into what EGFR does. So once you have ATP binding, so you'll initially have a growth factor bind, then you'll have EGFR dimerization. And then once you have ATP binding to the intracellular pocket, it then activates downstream proteins, which then triggers a cascade of events, which results finally in tumor growth and metastasis. Now, amongst the downstream proteins that get activated, KRAS is one of the downstream proteins. And what happens, and as a defense mechanism in the cancer cell, KRAS gets mutated to mutant KRAS. Now, typically in a normal cell, KRAS, or in a normally functioning cancer cell, KRAS requires EGFR to activate it. But in a mutant KRAS cell, it has found it's got its ability to turn itself on its own. So it really does not need EGFR activation anymore. And the cancer cell is already switched on and already multiplying and proliferating. And so it basically, in mutant KRAS cells, you basically left the EGFR inhibitors being completely ineffective. And so there is a lot of need for getting new drugs for mutant KRAS expressing non-small cell lung cancer. Right now, standard of care with the clinical guidelines are EGFR inhibitors are the standard of care for non-small cell lung cancer. Now, if you have mutant KRAS positive non-small cell lung cancer, you have much fewer options. And so with that, so what are the current strategies to overcome resistance? And so there are strategies where they've looked at specific, like now targeting the triple mutant. But what's going to happen after you target the triple mutant is it's going to go along the lines of HIV, where you then start having quadruple mutants and so on and so forth. So it's always going to be a moving target. And I don't think that is the best approach. It's also going to be very narrow spectrum. Another strategy that could be used would be just to target. So if mutant KRAS is the problem, you just make mutant KRAS inhibitors. But there are different types of mutant KRAS. And so you have different mutations. And though there are drugs that are being investigated, nothing has been approved so far, but there are drugs, but they target very specific mutations. So again, mutant KRAS is going to be another moving target, just like mutant EGFR. And so these are not the best approaches. Additionally, there are no drugs approved right now to treat, or no EGFR targeted drugs for either mutant EGFR, which has the triple mutant, or mutant KRAS cells. Now, there are literature reports which show that you can use EGFR inhibitors with another drug or another molecule that targets Aurora kinase. Now, aurora kinase is another kinase. It's a serine threonine kinase. It feeds into the EGFR pathway, but it feeds into it downstream of mutant KRAS. And so even if KRAS gets mutated, aurora kinase can still shut down that pathway. Additionally, if you give EGFR inhibitors, so the existing inhibitors, erlotinib and osimertinib, if you give them together with aurora kinase inhibitors, you'll have synergistic effects in cells. So this is in normal cancer cells that have no mutations. If you give EGFR and aurora kinase inhibitors together, you can have a more pronounced effect of tumor cell inhibition. Second point is that if you give EGFR and aurora kinase inhibitors together, you can also inhibit mutant EGFR non-small cell lung cancer. So you can inhibit cells that express this triple mutant. And the third point is you can inhibit cells that have this mutant KRAS in them as well. So you have now, with the combination of EGFR and AURK, you can target 3 mechanisms that is superior to existing EGFR inhibitors. And so looking at that, I tried to then see what I could do as a synthetic medicinal chemist to make this, to kind of capitalize on this. And I guess capitalize is not the right word, but how could I thought there was a need here for drugs that could be doing EGFR and AURK inhibition, but instead of giving 2 drugs separately, why not give 1 molecule, design a multi-targeted EGFR AURK inhibitor. So it will selectively target EGFR and AURK and will have no effects on other kinases. And so the reasons or the advantages of doing that is you would still have the synergistic EGFR AURK inhibitory effects in normal cancer cells, so which don't have mutant EGFR. You would have effects in mutant EGFR cancer cells, as well as in mutant KRAS expressing cells. So that is a significant advantage. And an additional advantage would be you would not have the disadvantages that we see with 2-drug regimens. So when you're giving 2 drugs separately, the 2 drugs can have interactions amongst themselves. You will have to time the dosing for the 2 drugs. And so you'll have to find the optimal timing to dose both of them. And so those considerations would not be necessary for dual targeted EGFR AURK inhibitors. And so then the question arises, are there dual targeted EGFR AURK inhibitors? That's the first question. And then there's another question, are there any dual targeted kinase inhibitors? And so there are dual targeted kinase inhibitors. There are many that have been approved, and you can selectively target your target kinases and not target the entire kinome that uses ETP as its normal substrate. And the second thing is there are no dual targeted EGFR AURK inhibitors that have been reported thus far. So I have looked up the literature, maybe people are working, but they're not publishing on it at the moment. And so this is really a niche area, which I think is an area where we could, grow. And so this was something that, I've been working on this for a few years. And so I was able to identify. So first then, moving on to keeping to my slides. So feasibility. So, it's a very nice idea that you would target EGFR and AURK, but can you really do it? And so to determine if you can really do it, you'll have to look at the pockets and see if you can actually identify a molecule that will fit optimally in those 2 pockets. And to the same extent, and actually be dual. So, at least in the 1 to 3 range of inhibition for both of these targets with no inhibition on the rest of the kinome. And so I did that. So I use a molecular modeling software, molecular operating environment, where I looked at the crystal structures for EGFR and aurora kinase. And then I looked at the pocket and I noticed that you can design molecules. There are many similarities. So these are the different regions. And in the interest of time, I'm not going to go over all of them. But there's a lot of backbone for the hinge regions that you can overlap, and it's identical in EGFR and AURK. There are many residues in each of these pockets. So if you have hydrophobic or non-polar amino acids, they overlap with the same place in aurora kinase. You also see that for some of your polar amino acids as well. So you can actually selectively target EGFR and AURK and not target the rest of the kinome. And you can make 1 molecule that does both. And so after having done that, so this is something that I did in my lab. I haven't disclosed the structures in any of my presentations, just because I do have a provisional patent, but I'm still protective till I get a full patent on these molecules. So there are no molecules that are disclosed here. But we've basically rationally designed molecules using structure-based drug design. The next approach would be synthesis, so solution phase synthesis, solid phase synthesis, and organometallic reactions. That is what my lab, those are our strengths. And so we've used a lot of that. We've made series of compounds. We have compound purification. So we have capability to purify our compounds with flash chromatography. We have access to a semi-prep HPLC at the Shimadzu Research Core, which I aim to use. I haven't had a need for it thus far, but it is available. Compound characterization for identity and purity requires NMR, proton as well as carbon NMR, and this is done through the Shimatsu Research Corps, followed by high-res mass analysis and elemental analysis, which is done externally to determine compound purity. So you confirm identity and confirm purity. Once we confirm that, we then send out the compounds to a contract research organization for enzymatic evaluation for EGFR and AURK. They have all the mutants for EGFR. And so there's 3 different mutants that I'm looking at, and then they have different versions of AURK. So you can selectively target AURK A or AURK B, and they have different applications. I haven't gone too much into it here, but you can, and we test them. So it's almost 6 kinases that we actually test our compounds against. Selectivity, so selectivity against a kinome panel is another step that we take. And then anti-cancer effects in cells are evaluated in collaboration with Dr. Amisa at Ferris. And so what are our results? And so without going into the compounds, what we have demonstrated thus far is we have been able to design and synthesize and demonstrate dual EGFR on AURK inhibition in our molecules. So we've demonstrated compounds that can be dual EGFR and AURK. These are nanomolar to single-digit micromolar in their inhibition. As part of the advanced grant, we've actually gone further where we have additional molecules which are nanomolar for EGFR and AURK both. We've also along the way identified selective nanomolar AURK inhibitors. So they are not dual, but they're selective for AURK. We've also identified new molecules that are selective and nanomolar for EGFR. In our hands, we've consistently seen better EGFR inhibition compared to AURK inhibition. I don't know how I'm doing on time. Okay, and so along the way, of course, all of our molecules are novel. So there's novel composition of matter that is being developed. Selectivity. I have made, I wouldn't say library because I think that's used too loosely, but I have made series of compounds, several series of compounds. And within there, we have tested selectivity for 3 of them. And we have been able to, we started out with a compound which was not very selective. It hit 24 other kinases, but we have been able to narrow it down to a compound now that exclusively hits EGFR and AURK at our tested concentrations and does not hit any of the other kinases in the panel. We have gone on now, so we've identified dual EGFR AURK inhibitors and we have selective compounds. We've gone on to test them in collaboration with Dr. Amisa at Ferris State University. Now, it was good to know that the compounds had single-digit micromolar inhibition in cell, but single-digit micromolar is not potent enough for future commercialization. And so the current efforts through the advanced grant are to improve potency for the compounds, which would then translate to improved potencies in cells as well. And so future directions involves molecular optimization and then subsequent commercialization efforts through a subsequent kickstart, if at all. But right now, the advanced grant focuses on molecular optimization. And so who has done this work? So my research team, and so these are the students who've been working with me prior to COVID, and they've come back after COVID hit as well. And so this is in their COVID, with the safety. They have been, so Aiden completed a summer research fellowship with Dr. Rivera, and he's very good with molecular modeling, and he was interested in continuing with research. And so he joined our lab in 2019, fall of 2019. And he does molecular modeling for all of our compounds. Sarah LaForce, as well as Kyle DeYoung, are both joined my lab as volunteers. And then they've been working through paid positions following the advanced grant. And then they mainly do the synthesis as well as compound characterization. So what are the next steps? So molecular optimization to improve potency of my molecules are the next step. So there's 2 approaches that I'm taking for that. The first is to just improve potency with better binding interactions in EGFR and AURK in the active site as well as the allosteric site. Another approach that I'm taking is to just improve the drug-like properties. So my molecules are highly nonpolar, and nonpolar molecules don't penetrate cells very quickly, and so, or very well rather. And so I'm now working to make them more polar, and so that they can penetrate cells. So that whatever I'm seeing for the enzymatic assays, that nanomolar inhibition can translate into the cells as well. So with that, I'm going to end. So the acknowledgements would be to my students, who have completed all of this work and, who've been stayed true to the project, despite, some extenuating circumstances with the pandemic. Collaborative support. So without Dr. Amisa, I really don't think I would have even been eligible for the advanced grant. Dr. Amisa actually demonstrated he has cells which have mutant EGFR as well as mutant KRAS, so the resistant lung cancer cells. And so he was able to demonstrate for me that my compounds are effective in those mutant lung cancer cells, which was, I think that really, again, similar to the advanced grant, that really took my project into a new league and found my niche. The Shimadzu Research Corps for all of their instrumentation consistent support with keeping it, working and running, and their patience with me. And then funding support, again, I'm going to reiterate this, advanced grant has been critical in helping me with the data from the advanced grant. I was able to subsequently get an NIH grant. I got the R15 REAP, which is going to be used extensively for molecular optimization and improving. And that grant is collaborative with Dr. Amisa as well. So we're still in the same area. And then institutional support. So the College of Pharmacy, of course, for space, equipment, and support whenever needed. Dr. Dowling from the Office of Research and Sponsored Programs, who actually gave me the initial encouragement to apply for the advanced grant and kind of believed in the commercialization potential for my project. And then also and Ferris State University overall, of course. And then I'm also supportive of or appreciative rather, of the intellectual property application, and commercialization efforts will get expensive. And I'm really appreciative of Ferris committing to that for us, for all three of us rather who spoke today. So with that, I'm going to stop and I'll take any questions.
Thank you, Sonali. Thank you, Eric and Daniel as well. We have just a few minutes, maybe right around 5. If anybody has any questions, we just have to allow for time to transition for the next room. So if you have questions, go ahead. Eric and Daniel have both put their emails in the chat as well. So if anybody wants to email them with questions, and I'm sure Sonali will do the same. You guys are getting kudos for many. Yep, Sonali's email is there, and then a lot of great presentation comments as well. So
we're learning the language of emojis for appreciation and applause during these Zoom presentations.
Yeah.
I refuse to make that step.
Oh, I haven't seen the congratulations one there with the.
There's going to be a TikTok dance at the transition of the hour.
Excellent. And Eric, I'll meet you at the mailbox later today to celebrate, okay? Eric and I are neighbors.
Daniel is the greatest neighbor. I put the whole neighborhood to shame because he keeps his house great. I'm the ne'er-do-well, but we have a great time.
Props to you for using the word ne'er-do-well. I thought that was one that only I trotted out every now and then.
I'm one of those scalawags and carpetbaggers.
You mountebank.
Great presentations, everyone. I was wondering if you wanted to talk or reflect a little bit on the role of the T3N mentor that has helped along the way. I think we've all benefited from Karen Studer-Rabler, who is kind of the silver lining here too. Anyone want to reflect on how that's worked and how important that's been?
Well, the T3N mentoring network is at no cost to Ferris, and I've spoken with folks that had 30 years of experience in the drug industry. Bruce Auerbach. There's really too many mentors to name in this short time, but Bruce Auerbach really got us on the right track, because sometimes you have an idea and you don't know where the commercialization space is. They helped us to start thinking about the antibiotic development space, which is more of a blue ocean than a red ocean. Really, their vision and guidance, we wouldn't be here today if it hadn't been for them and the resources they provided. So they've been invaluable.
I second that as well. Karen has been very encouraging. I do need encouragement sometimes. I really think that my compounds are not ready for commercialization. I kept telling her that, and she convinced me that they are and what I can do. And Bruce, Dr. Markham did the same. And so I really appreciate the encouragement, as well as, the next steps, because I think sometimes, if you think of commercialization, that means, you're finally going through FDA approval and all of that. It seems overly ambitious sometimes. And so and it can be intimidating. It was for me at first, but I think I'm on board with, how that can be done. And she asked me to enroll in the fast-paced program as well. They're always available. They're happy to review your grants for you too, when you send out your application. So all of that she has been very supportive. And, I'm sorry I've missed acknowledging her in my slide, but, she was a big part of, the success on the project. Despite the pandemic, we kept meeting, we kept talking about what we could do. So I appreciated all of that.
And I have very little to add except to acknowledge Karen's role as that person who will kick your butt and get you back on track. We're all very busy with a lot of different responsibilities. It's nice to know there's someone who's checking in on you every month. So she is certainly responsible for my particular project. I know I can speak for it, being as developed as it is at this point.
Great to hear. I'll make sure Karen receives your feedback. She's an awesome person to work with. And, you know, there are two sides of the coin. There's the mentor and the mentee. And if the mentee doesn't want to be the mentee and doesn't want to take advice from the mentor, then we kind of have a problem. So kudos to you for listening and being coachable and, you know, grabbing onto that baton as she hands it to you, or maybe beats you with it. And, you know, keep working based on everything else that you're doing. So kudos to you as well.
All right, thanks again to our presenters and everyone, and this will conclude this session, so you have time to move on to your next one.